The cell cycle is the series of events that a cell goes through as it multiplies and divides. And one part of the cell cycle is what we refer to as mitosis, which is the production of two genetically identical cells. So we are continuing on our journey through chapter 10 in the book, and we are on section 10.2 on page 280 if you are following along. So we're going to be looking at the entire cell cycle, which produces two daughter cells, two genetically identical daughter cells from the main cell. So if we look at all of the phases of the cell cycle, it starts with G1 phase, then proceeds to what we call S phase, G2 phase, and then M phase, which is known as mitosis. And this is the complete cell cycle. This is also found on page 281 in your book. If you wanted to pause the video right now and draw this cell cycle out or sketch it, you could in your notes. We will also be making this chart wheel next week in class. So if you don't feel like sketching something real intense, it would not be a big deal. But you may just want to draw this out in your notes as a way of taking notes as we go through what each part, what each phase means. So the phases of G1, S phase, and G2 make up what we call interphase. So G1, S phase, and G2 make up the part of the cell cycle that is known as interphase. It's kind of the in-between period of growth before actual division of the cell. So during the G1 phase, cells do most of their growing during this phase. They will increase slightly in size and make new proteins and organelles. The G stands for gap. Gap 1 is what this is actually called, where we have a lot of cell activity and growth going on during these phases. So G1, they do most of their growing. They increase slightly in size. They make new proteins and new organelles. From there, the cell then moves on to the S phase. The S in the S phase stands for synthesis because this is the synthesis of DNA. This is where the DNA is replicated. Replicated means to copy. So this is where the chromosomes, which are your DNA, the DNA is copied. So at the end of the S phase, a cell actually has twice as much DNA as it did in the beginning. So here the amount of DNA doubles. Then into the G2 phase, otherwise known as gap 2 phase. Okay. This is the shortest and quickest of all of the different phases. A lot of the organelles and things that are needed for the actual division, the actual splitting of the cell into the two daughter cells are produced here. So a lot of the organelles and molecules are made here that are going to be needed for the actual division of the cell. This is the final preparation for cell division when the cell enters into mitosis. So interphase, the in-between phase, contains G1, S, and G2 phases. This is the preparation for actual cell division, and probably the most important thing that occurs here is the replication of the DNA, where the genetic information is copied so that it can be passed on to the two daughter cells. So from here, it will enter into mitosis. And mitosis actually has 4 major phases known by the abbreviation PMAT. PMAT stands for prophase, metaphase, anaphase, telophase. And then the fifth and final part is cytokinesis. So you've got prophase, metaphase, anaphase, and telophase. And we're going to look at these each individually. The first one is prophase, which is the longest phase. So here, the genetic material that's in the nucleus of the cell will actually become visible. Prior to that, you can't see the genetic information that's inside the nucleus. You can't see the DNA. But during prophase, it coils up and becomes very visible in the nucleus. The other things that you have happening here is the centrioles will move to the opposite poles. The ends of the cells are called the poles, sort of like the North Pole and the South Pole. The centrioles will move to opposite poles. These long spindle fibers will also form. And towards the end of this phase, the nucleus, the nuclear membrane, or nuclear envelope will break down as well. So again, a few things taking place here. Chromosomes coiling up, condensing, and are now visible inside the nucleus. Centrioles move to the opposite poles. You've got the spindle fibers that form and the nuclear envelope that breaks down during prophase. Then the next phase is metaphase. Metaphase is very short, only lasts for just a very few minutes. When this point in time, the chromosomes will line up in the middle of the cell. They line up on the equator in the middle of the cell and attach to these spindle fibers. In the middle of each chromosome is what is called a centromere, and that's the part that attaches to the spindle fibers. So during metaphase, the most important part is that the chromosomes line up in the middle, attaching to the spindle fibers, and they're attached at what is called the centromere. This is a chromosome that is duplicated, because right now the cell has twice as much DNA as normal. So you have 2 copies of every chromosome. After metaphase, move on to anaphase. At anaphase, the sister chromosomes or sister chromatids will separate. So those duplicated parts separate. The duplicated chromosomes will separate into the individual chromosomes. So they separate and they move to the opposite ends, to the opposite poles. So one chromosome went to this pole, the North Pole, and one went to the South Pole, so to speak. So they split, the sister chromatids split and separate into individual chromosomes at each pole during anaphase. So the key point of anaphase is that during anaphase, the chromosomes separate. They move along the spindle fibers to the opposite ends of the cell. The next phase is called telophase. At this point, the chromosomes will begin to uncoil, so to speak. The nuclear envelope will now start to reappear, but now you will have 2 of them. The spindles will also break apart. And so the nucleus is going to reappear. These chromosomes will actually begin to uncoil so that you will not be able to see them anymore. And you have the 2 nucleuses during telophase. Now the actual process of mitosis is complete. PMAT, prophase, metaphase, anaphase, and telophase. But at this point, the cell has not completely split yet. It has what is called a cleavage furrow right here. Okay? but it has not completely pinched in half. So you've got the two nuclei, but you've only got one cytoplasm, one cell at this point. So now cytokinesis must take place. And cytokinesis is the actual point where the cell pinches into two. So the cytokinesis pinches the cell in half, and you have two daughter cells that are exact duplicate copies of the one parent cell. So you've got a new cell membrane that has formed. It has pinched. The DNA was copied, so both these cells are identical to the original single parent cell. In a plant cell, it would not pinch off. There would be a new cell wall that would form here. So there would be a cell wall that would develop and form two plant cells. Where in cells that don't have the cell wall, a cleavage furrow develops and it literally pinches off right here in the middle. So on page 285 in your book, you can kind of see the interphase, which has G1 S phase and G2 phase. And then you can see mitosis, which has prophase, metaphase, anaphase, and telophase. And then the final part, which is cytokinesis, which has the actual pinching in half of the cell to make two identical daughter cells. You will also notice on page 284, you will actually see the difference between an animal cell and a plant cell, and how the animal cell pinches in half, where a plant cell will have a new cell wall that develops in between the two of them called a cell plate. Regulating the cell cycle. There is, as we talked about before, these checkpoints that a cell goes through so that there's not uncontrollable cell growth. And it actually can shut off cell division, so the cell cycle can be regulated. They're called cyclins, and they are proteins that regulate the cell cycle so that it happens or doesn't happen when it needs to. So those things are referred to as cyclins, regulating the cell cycle. There are both regulatory proteins both in and outside of the cell. So you have internal and external regulators. Internal cell regulators will prevent the cell from growing and dividing if a step has been missed. Like, for instance, if the chromosomes don't get duplicated, then the internal regulators, the internal cyclins, will prevent the cell from growing and dividing. Where external regulators respond to external events to stimulate growth. And these are very important during like wound healing. When you have a cut or something, the neighboring cells around the cut will trigger the cells to grow and divide rapidly for healing. So there's internal and external regulators that can speed up or prevent growth of cells, prevent growth and division of cells. So that should sum up mitosis and chapter 10.